Hello everyone. Today I'm going to be talking about genetics, someone's favorite topic, I'm sure. Uh, I'm going to be going over summarizing what you would expect to learn in a typical college course when dealing with genetics. I'm going to be going over a brief summary of that, as well as getting into a few genetics problems and solving for them. So the first thing that we're going to be talking about is population genetics, which is what most people are familiar with. This is where you have one gene with two alleles. And the example that we're going to be using are snapdragons. Snapdragons come in a lot of different colors. For this example, we're just going to pretend that there are only two colors. We'll say red and white. And we'll say that the big A allele codes for red, and the little a allele codes for white. So you know that the genotypes for these would be big A, big A, big A, and little a, little a. And if you remember, these would be homozygous. They have two of the same allele. These would be homozygous. And this right here is your heterozygote. And I'm going to talk briefly about genotype frequency versus allele frequency. This is something that people get confused with. So let's say that within a population, 50% have this genotype, 30% have this genotype, and the remaining 20% have this genotype. And it doesn't matter whether or not there's complete dominance or incomplete dominance here. That would be the phenotype. The phenotype doesn't matter when dealing with genotype frequency. 50% would be big A, 30%, and 20% would be white. So these directly tie to genotype frequency. However, allele frequency would be different. Let's say that this is a population size of 100, and since each individual flower will exhibit two alleles, since they all have a genotype, that means that there would be 200 alleles. So, using our population size of 100, we could say that 50 have the big A genotype, which means that each individual flower has two capital A or red alleles, which means that just from these 50, there are 100 A's. Then we move over here to the heterozygotes, where 30 have a genotype of big A and little a. So what we could do here is we wouldn't multiply it by 2, since each flower only has one big A, we would just add 30. And then of course solving for little a, since 20 flowers would have little a, we could say that there would be 40 little a's coming from the homozygotes for the white, and then 30, oops, didn't do that quite right, 30 as well coming from the heterozygotes. So the allele frequency in this particular case for the red allele would be 130 over the population size of 200. I'm sorry, not the population size, the allele population size. So 130 out of 200, and little a would be the remaining 70 out of 200. So this just means that out of the 200 alleles present in the population of 100 flowers, 130 of those alleles are red, and 70 of those alleles are A. And you could also put this into a percentage if you wanted to. In this case, it would be 65%, and this would be the remaining 35%. So you could say that 65% of the alleles in the population code for red and 35% code for white. So now that we have that out of the way, it's pretty basic. And then of course we can do a genetics problem. With that, we can say if you breed a heterozygote with a white flower, what will be the genotype frequencies, or what would you expect to be the genotype frequencies for the offspring? And of course for that, we draw ourselves a nice Punnett square. And we should all remember how to set these up. They're really easy, really useful. And you just fill those out. So this tells us that out of 4 offspring, 2 would be heterozygotes and 2 would be homozygotes for the recessive allele. And you can see here that the genotype frequency would be just 2 out of 4 for both of them. So the genotype frequency would be 50% heterozygotes, 50% homozygotes for the white. Allele frequency itself, you can see would actually be, for the red allele, would be just 1 out of 4, or in this case 2 out of 8. So 25% red allele, and the remaining 75% would be the white allele. So again, very basic stuff. So now we're going to start to get into more complicated things, but again, we're just going to be kind of summarizing them. So next thing we're talking about is the Hardy-Weinberg equilibrium. This is a very useful equation, and I should note before we get into this, if the allele frequencies are changing in a population, the population is not in Hardy-Weinberg equilibrium. Now that being said, this equation is still useful and usually fairly accurate for population sizes. But again, if it can't produce completely accurate results if the allele frequency is changing, or if evolution is taking place. But the Hardy-Weinberg equation is p squared plus 2pq plus q squared equals 1. Now, what the heck does that mean? This gives us genotype frequencies, whereas the individual variables can be substituted for allele frequencies. But when you actually solve for it, it gives genotype frequencies. So this is really useful. So let's do an actual example here. Let's say that we have a reindeer herd, and there is an allele that selects for red noses. Christmas is coming up. It's a good example. So there are alleles that cause a red-nosed condition, and it's very rare. Out of let's say our population size is 10,000, and let's say that 25 out of the 10,000 individuals have red noses. What would be the predicted frequency of heterozygotes in this population? That is the question. What would be the predicted frequency of heterozygotes? We know that 25 have the red nose, and this is a recessive condition, so that means that the 25 are homozygous for the recessive allele. So how do we use this equation and this information to find heterozygote frequency? Well, what we would do is the 25 out of 10,000, I've already put it into fraction form. That gives us a genotype frequency for the recessive allele of being 0.0025. And as I said before, these values right here will give us genotype frequencies. So if we say that Q is for the recessive allele, Q squared will give us the homozygous recessive allele genotype frequency, which we actually just found right here. So if Q squared equals this number, then the square root of this number will actually give us the allele frequency for the recessive allele, which if you solve that comes out to be 0.05, or 5% of the population carries this allele. And then we can solve for that and say that the dominant allele would then be this right here, P. P would be the remaining 95%, or 0.95. So if we know the allele frequencies for P and Q, we can solve for any genotype. This is what we want right here, because this will give us the heterozygote frequency. So 2 times P, and in this case it's not the allele frequency of 0.95, so 2 times P times Q actually will give us the heterozygote frequency, which comes out to be 0.095. So 9.5% of the population will be heterozygotes for the red nose condition. So fairly simple, yeah, fairly simple. So let's move on to something else. And right now we're going to be talking about S. S stands for selection coefficient. And now we're getting into more real life scenarios, more of the fun stuff. S stands for selection coefficient, which means a loss of fitness for a particular genotype. And in this case, fitness, we're not talking about like a really muscular elk or something. We're talking about the actual reproductive success of an individual with a particular phenotype. And in this case, we're dealing with relative fitness. So the max fitness is 1, the lowest is 0. If a fitness is 0, we say it's lethal, because they can't reproduce if they're dead. So we're dealing with relative fitness here. So one of the genotypes will always have a fitness of 1. So S would just be the 1 minus the fitness, and fitness would also be 1 minus S. So I'm actually going to show you a good picture, so you guys can just picture this a little bit in a real world scenario. So let's say that you have a recessive allele, where here it says the selection coefficient is 0.5 against a recessive allele. So that means that the fitness is 0.5, or another way to word that would just be you're 50% more likely to die or to not reproduce if you have this particular genotype right here. The other two, again, have a fitness of 1, and the selection coefficient here would be 0.5. So the dominant allele is what is being selected for. So it gives us a nice little chart here, and it shows us that even if the frequency of the dominant allele starts way down here towards 0, and the frequency of the recessive allele starts towards 1, if there is a selection against the recessive allele, you'll see this exponential curve of the frequencies. So the frequency of the recessive allele will go down very quickly, and then over time will just keep getting closer and closer to that 0 value. the frequency of A will grow extremely quickly and then again kind of average out. And if it gets all the way up here and actually gets to 1, we call that fixation. An allele becomes fixed when the allele frequency is 100%. Now let's move to the opposite of that. Let's say that there's selection for a recessive allele and against a dominant allele. And here the selection coefficient is even greater. So we see that the dominant allele has a fitness of 0.4. It means it's only 40% as likely to reproduce as this genotype with the recessive allele. So again, the frequency of the dominant allele starts towards 1. The frequency of the recessive allele starts towards 0. And it really takes a while for things to catch on. But once it happens, it spreads very quickly. The allele will. You see that exponential growth again, and it'll happen slow at first, and then quickly later. So that's the general overview for that. So moving on to something else that you might learn in a college class would be actually finding allele frequencies for the next generation. And this might sound like a really hard thing to do at first, but we've actually got a few equations that are great. And I'm actually just going to go straight from my outline here. Not a very big budget in this, but that is okay. So we have 4 equations here, each relating to a different scenario. So for example, if there is a completely dominant advantageous allele, you would use this equation. If there's incomplete dominance for an allele that is advantageous, you would use this equation. If the heterozygote has a higher fitness than homozygotes, that's called overdominance, you would use this equation right here. If the heterozygote has less fitness than both homozygotes, then again you would use this equation. And you notice this right here, this is the key, delta P. That is the change of the allele frequency. That's not the new allele frequency, that's just the change. So that'll get added to the old allele frequency to get the frequency for the next generation. So you want to use these for reference. Hopefully, if you have to learn this in class, your professor won't make you memorize them. So let's do a problem with them. So using an example from before with the same red-nosed reindeer, let's say that red-nosed reindeer get shot a lot more. They get shot actually 20% more. So let's just draw this out. We have PP, PQ, and QQ. This will be for normal-nosed. This will be for pink-nosed. This will be incomplete dominance. And this will be for red-nosed. The red-nosed are about 20% more likely, or less likely, I'm sorry, to survive. So their fitness would be 0.8. Heterozygotes, which have pink noses, they are about 10% less likely to survive. So they would have a fitness of 0.9. And of course, the regular nose would have a fitness of 1. So again, the dominance here would be incomplete dominance. So we'd go back, you would refer to the equations that we just talked about. And for this particular scenario, you would use the equation delta P equals SPQ all over 2 times 1 minus SQ. And this is why we talked about what S actually means, is because it's useful in these equations. So now we are solving for delta P, and we plug in our S value, which we're going to take this right here. This is our S value. Since we're solving for the frequency of heterozygotes, we're going to use this as our S value right here. And you might be wondering, well, what about this number? This one, we'll get to this later, but this one's actually your T value. And this is the same thing again, selection coefficient. It's just distinguishing between these two. So we're going to use this as our S value since we're solving for this particular allele. So we're going to have delta P equals, and then this will be 0.2 multiplied by P, which is the allele frequency, which again we already solved for, if you can remember that. But remember, we found a P value of 0.95, and then multiplied by Q, which we found that as well as being 0.05, all over 2 multiplied by 1 minus S, which again is 0.2, and multiply that by 0.05. And if you do the math, you get delta P as equaling 0.0048. And again, this is the change in the frequency, not the new frequency. So if we're solving for what is the frequency of heterozygotes in the next generation, we're actually going to add this to the old generation. So this would be, in this case, the 0.0048 value that we just found. That's not an 8, but that is okay. Plus 0.95, which gives us a value of 0.9548. And this, again, this isn't the frequency of the heterozygotes. This is the new P frequency. So, going back to good old Hardy-Weinberg equilibrium, remember how to solve for heterozygotes. It was 2pq. And we know 2. We know P. We just solved for it, the new P value. So instead of 0.95, it's 0.9548. And that leaves Q, which instead of being 0.5 will be 0.0452. And we got this number just from doing 1 minus the P value, because these two numbers have to equal 1. So if you do the math here, this is again is the Hardy-Weinberg, from the Hardy-Weinberg equation. This will solve for heterozygote frequency, and this will give us a final value of 0.086. And if you remember from before, this is a lower value than before because heterozygotes have a small disadvantage. They're 10% less likely to survive. So let's clear that and move on to the next thing, which is long-term equilibrium frequency. And this basically just means this is what the allele frequency will move towards over time. We already mentioned before, if an allele frequency reaches 100%, we say that it's fixed. So again, it's some more math. I'm sorry. We solve for long-term equilibrium frequency with this guy right here. We call him Q hat because he's got a little hat on him. So Q hat will equal the square root of U over S. And we already know what S is. This is the selection coefficient, or loss of fitness. But what is U? U is actually the mutation rate. And you might be wondering, well, how the heck do you know that? Well, this is not an equation that we're going to be using too often, so I won't get into it too much. This equation can actually only be used to find the equilibrium frequency of rare, harmful, and recessive alleles. So again, we won't use this very much. Only in cases of rare harmful recessive alleles is this equation useful. But let's look at a genetics problem where we can use this. Let's say that there's a lethal allele that has a mutation rate of 10 to the negative 5. That's the mutation rate. And we could say, what is the equilibrium frequency if this is the mutation rate? Well, we say q hat equals the square root of 10 to the negative 5. And do we know what s is? Well, we said that it was lethal, so that means that s would just be 1. So when you plug in the numbers and do the math, you actually find that q hat will be 0.0032. And this is allele frequency again. So this doesn't mean that 0.3% of, let's say this is in people, let's say this doesn't mean that 0.3% of the people will have, will die from this. This just means frequency is 0.32%. So 0.32% of people will carry this allele, whether it be in a heterozygote, which is more likely, or in a homozygote, which would, again, be lethal. So let's look at a more practical q hat equation, one that is a little bit easier to find. This one will be q hat is equal to t all over s plus t. And again, we said that t is just the other value for the other selection coefficient. But you got to make sure you can't get confused, you can't mix them up when you do problems with this, and I'll show you how to do that. So again, let's go back to some snapdragons. Let's say that's, all right, let's draw this out actually. Let's again go a, little a, and little a, little a. Let's say that pink snapdragons have a fitness of 1. Red have a fitness of 0.75, and white have a fitness of 0.9. So what we want to do is find the long-term equilibrium frequency for the red allele. Well, that's the genotype, the red allele. So what we would do is we would go q hat equals. Now which one do we use as s and which one do we use as t? And the way you remember this is whichever one you're solving for, that is your s value. So we're solving for big A. So we're going to use the one under big A as our s value. And then this guy right here will be our t value. And if we were solving for the long-term equilibrium frequency of little a, this would be our s value, and this would be our t value. So we can just plug in these numbers. And again, we don't plug in 0.9 and 0.75 because this is fitness. We plug in the selection coefficients for each of these. So it'll be 0.1 because 0.9, you get 0.1 from that. And then 0.25 plus 0.1, which gives us somewhere around 0.29, which means that the long-term allele frequency for the red allele would be somewhere around 30%. And it should be noted that selection usually won't drive a dominant allele to fixation, because recessive alleles can easily get carried in heterozygotes. So if q hat is 0, it's unlikely that it will happen that way, unless the allele is of course lethal and dominant. So now we're actually going to get into the second part of genetics, which fortunately for us has less math in it. And this is called quantitative genetics. And again, going back to my outline that I'm using, This is a very important part of quantitative genetics. Quantitative genetics isn't just one gene with two alleles. This has multiple alleles coding for a phenotype. So you'll get this bell curve with whatever it is that you're looking at, whatever phenotype it is. You're going to get a bell curve if it's in quantitative genetics. So let's talk real quickly about selection. So there's a few different types of selection. One would be disruptive, which means that the average is bad. So if we have a little bell curve here, the average is bad right here. You don't want to be that. So what will start to happen there is the bell curve will start to split into two different directions. Instead of this, you'll get something like this, which, as you can imagine, or if you can picture it, this is a great way for a new species to show up. There's also stabilizing selection, which means that the average of the bell curve is good. So your bell curve would end up looking something like this, with not a whole lot of variation if the average is good. Have directional, which means that one extreme is better than the other, and that would just move the entire bell curve. It would just shift it in the direction of that extreme. So what are some quantitative genetic traits? This would be things like IQ, height, weight. Again, anything where you'll see that bell curve. That is a good sign that it's quantitative genetic traits. So with quantitative genetic traits, we're going to talk about variance, something very closely related to it. So variance, we're going to give, unfortunately, a little math here. So this is our equation. VP equals VG plus VE. Now, what does this mean? This means variance. All these V's stand for variance. So the variance of a phenotype in a population, and this is what is actually measurable. So like IQ, you can measure IQ, height, you can measure height, you can measure weight. This is variance of phenotype in a population. VG stands for the variation due to genetic differences within a population. And VE is the variation due to environmental differences in a population. And you'll notice I'm saying in a population. It's important to note that this applies only to populations, and you can't actually say this for individuals. So for example, if I'm, say, 150 pounds, I can't say that 130 of that is due to my genetics and 20 of that is due to the environment. It just doesn't work like that. You can't apply it to individuals. So with that in mind, let's move to heritability. And heritability, it's a common misconception. Heritability, it's not just how genetic something is. Heritability has a lot to do with a correlation between the average phenotype of parents and the phenotype of the offspring. So let's draw ourselves a little bell curve here. That was a good one. And that's average, right at the top of the bell curve there. That's average. So with heritability, let's start off by saying that directional selection can actually produce phenotypes outside the range of the original species. So for example, something like this. This is a wild banana, not something that I would be too excited to eat. The phenotype for this one, let's say that this phenotype is the size of the banana. The ones that we eat are somewhere like way over here, somewhere outside the original range of the phenotype. And that's due to directional selection. I mean, humans have been breeding these things for years to be bigger and better and tastier. But so we're gonna use a few more variables here, unfortunately, for some more math. S and R. So I said before that heritability has a lot to do with a correlation between the average parent phenotype, which we just say mid-parent, and the offspring's phenotype. So the change between the population average and the mid-parent, let's say that the average of the parents is somewhere around here on the bell curve. This value right here is S. And the change, let's say they have an offspring, which is right here. So this value right here, yes, you guessed it, R. So this S actually stands for selection differential, and this R stands for response to selection. So with that in mind, we get to do some math. Woohoo! So heritability, H squared. And for some reason, it's H squared. Heritability squared, H squared just stands for heritability. But anyways, heritability equals R over S. And we just learned what those values are. Of course, this is the response to selection. This is the selection differential. And that's not the only equation. H squared, that's not a very good H squared equals VG, which we learned about, over VP. Now you can see, you can derive from this equation right here, especially that guy. If genetic variation is low, heritability is low, which means that if any selection happens, the species won't respond well to it. So if, for example, there's negative selection, if there's selection that reduces fitness and the species can't respond well to it, then it won't turn out too well for that species. So if an extreme is being selected for, there needs to be some a decent amount of genetic variation in the population. So no inbreds. Sorry, Joffrey. No inbreds. So the higher the H squared or heritability value, actually the more and faster change in phenotype between the parents and the offspring will be. If heritability is low, selection really has very little effect on change. So moving along with that idea, traits closely correlated to fitness will eventually exhaust their variation in the genetics, and they will exhaust their heritability. Their heritability will go down. And if they have been selected for a long time, again, the VG value is exhausted, meaning that really only environmental factors can change the phenotype. If you remember, the VG plus VE equals VP. And if VG is very low, all you have left is the variation due to environmental factors. So, and because, while we're on the topic of environmental factors, because environmental factors play a role in phenotypic variance and thus heritability, the H squared value found can only be applied to the population in that particular environment. So let's say elk in Colorado have a heritability value of 0.8. And again, heritability ranges from 0 to 1. If elk in Colorado have a heritability of 0.8 for their antler size, that value could not be applied to, say, elk in Utah because of the different environment. So let's look at a few different problems. Let's start with an easy one. Um, so we scientists in Colorado, I'll use Canada. Scientists in Canada found a heritability estimate for pig's weight. Would that study be useful for pig breeders in Utah? No, because of the different environment. No, that's not how it works. So, moving on to something a little bit more complex. And I'm actually just going to go back to my outline here, and we'll just read it straight from here. What is the variation, phenotypic variation for a population of elk with antlers sized 87 inches, 102 inches, 108 inches, 99 inches, 97 inches, 89 inches, and 100 inches long? So the way to actually find variation, the phenotypic variation, I didn't explain this equation earlier, but what you do is first you find the average, and the average of this population size is 97. And then, as you can see here, you measured and minus it by the average, and that answer you square and add it to the next value, and add that to the next value, and so on and so forth. And then you divide all of that by the population size minus 1. And that's how you calculate variation. So in this case, the variation we got, the VP value that we got would be 64.6. So moving on to something else, something with heritability. If heritability is 0.75, what is the VG and VE value? And this is using the elk population that we just looked at before. The VP value that we solve for will be useful in this equation. So we know that the heritability is 0.75. What is the VG and VE? Well, first we say, well, we have heritability and we're solving for VG, so we're using this equation. We already found the VP from doing that variation, phenotypic variation from the last problem, and we got that to be 64.6. And now we just solve for VG, which we find to be 48.45. So then how do we solve for the environmental variation? Well, we know that VP equals VG plus VE, and we know VP is 64.6, and that equals VG, which we just found to be 48.45, plus VE, which is what we're solving for. So then, of course, you just do 64.6 minus the VG value, and your environmental variance turns out to be 15.95. So that's how you solve for that. So let's move on to something else now. Let's say that 97 inches is the average antler size of some elk, and you breed two parents. The father has 105 inches, and the mother had 97 inches is the average, and you breed two parents with the mid-parent value is 107 inches, and we're going to be calculating for S. So we're keeping the same heritability value that we've had before, assuming that this would have to be in the same environment. We're going to calculate R, and we're going to use R to find the length of the offspring's antlers. So we found 97 to be the population average, and the mid-parent average was 107. So if you remember, S is just the difference between the midparent and the average. So it's just 107 minus 97, which gives us a value of 10 inches. So now we can solve now we can solve for R, because we know the heritability value to be 0.75, and if you remember, heritability equals R over S, which we just solved for S. So 0.75 equals R over 10. And R, which again is the response to selection, R would just be 7.5 inches. Now this does not mean that the offspring will have 7.5 inch antlers. This means that the offspring will have 7.5 inches above the average size antlers. So the offspring's antler length would actually be 104.5 inches. Now let's take that a step further and use this as the parent's value, 104.5. We'll use that as the parent value and find the antler length of the offspring. So we're going to have to find R and S all over again. So 104.5 is the midparent. And we're going to say that the heritability hasn't changed and the population average hasn't changed as well. So 97 again is population average, and this is the midparent value. So that's going to give us 7.5, which will be our S value. And we said heritability didn't change, so 0.75 is heritability is over R over S, which we just found to be 7.5. So then you just multiply 0.75 by 7.5 and solve for R. And we find that R equals 5.625. And again, this doesn't mean that they'll have 5.65 inch antlers. This means that the offspring will have this many inches above average. So we add that to the average to give us a total antler length of 102.6 inches. And you might be saying, if we're selecting 4 long antlers, why is the antler size slowly moving down? Well, in a real population, the average would be moving up along with it. So it wouldn't be 5.65 plus 97, it would be 5.65 plus whatever the new average is, taking into account, selection. So you'd have to find for equilibrium frequency, but over time, the antler size you could eventually get it, if heritability is high, you could eventually get antler size to be well outside the original phenotype values. I don't know if that's photoshopped. That's quite the antler size. But so again, if the selection is strong, the variation due to genetic differences will be used up, and this will actually reduce the response to selection. So as you keep selecting for larger antlers, the R value will keep going down, as well as the H value. And as selection favors an extreme, that population mean will move in that direction. So again, when the R value is added to that mean, it will eventually surpass the original limits for the phenotype being selected for. And here's a dog for Christmas. Merry Christmas. So there's a summary of genetics, what you would expect to learn in a typical college course. It hopefully wasn't too boring, but anyways, thank you for stopping by.